computer, right?
To start.
I'm going to start in Spanish. Okay.
No problem.
Buenas tardes. Agradecemos su presencia en esta magna conferencia. Ustedes ya han estado tal vez algunas veces aquí escuchando a la doctora Yonath, quien es Premio Nobel de Química 2009. Y no voy a decir mucho más de lo que ustedes tienen ya seguramente en sus manos, la semblanza. Solo quisiera hacer algunas precisiones sobre la doctora y sobre la importancia de su trabajo. La doctora Yonath nació en Jerusalén y una historia interesante que ustedes verán en la semblanza que tienen en sus manos, pero la doctora Yonath, a mí me gustaría decir que esto que está escrito en la semblanza hace una persona muy interesante, muy comprometida y un ejemplo de trabajo duro para llegar a lo que ella ha logrado. Yo he estado charlando con ella y ella me ha dicho que el premio Nobel fue un accidente en su vida. Ella estuvo trabajando porque desde el principio, una vez que terminó su carrera en química y después hizo una maestría y doctorado en bioquímica, ella prácticamente se enamoró del ribosoma. Y se enamora por una razón. Se enamora porque ella cree que la clave del origen de la vida está ahí. Hoy nos dará algunos datos sobre eso. Tiene hallazgos muy recientes donde me parece, en lo personal y comentando con ella, un hallazgo mayor en biología. Cómo se establece, cómo se cataliza el enlace peptídico en una estructura que le llama protorribosoma, que está desde especies muy primitivas hasta especies muy complejas. Como ustedes lo habrán leído ella ganó el premio Nobel por dilucidar por primera vez la estructura tridimensional del ribosoma, lo cual no era sencillo, fue muy complicado porque es una estructura supramolecular que debe estar mucho tiempo expuesta a los rayos X, y eso traía una consecuencia, su desintegración. Pensando, como solamente los genios lo pueden hacer, encontró en los osos polares una idea, cómo empacaban ellos estas estructuras antes de hibernar. Y eso le dio una idea y se desarrolló la cristalografía de rayos X a temperaturas de menos 185 grados, y eso le permitía tener estructuras estables y poder hacer la dilatación de rayos X, conectar los datos y dilucidar la estructura. Pero además, algo muy importante fue que con el estudio del ribosoma ella pudo entender y generar mucho conocimiento acerca de los blancos de antibióticos en el ribosoma. Y eso también es muy importante porque además nos platicará cómo los microorganismos desarrollan toda una guerra para matar a los demás, y el blanco es el ribosoma. Todos estos hallazgos, pues la llevaron al premio Nobel, y pues a partir de ahí ella lo dice que su vida no ha sido fácil, al contrario, ha sido más difícil porque ella ganó todo. Sin embargo, ha estado empeñada en seguir trabajando, y esto le ha hecho merecedora de varios doctorados honoris causa por varias universidades. Todo lo que les estoy diciendo está en la semblanza si quieren profundizar un poco más. Pero lo más extraordinario es que ella es una profesora extraordinaria de la UNAM, adscrita a la Facultad de Química, y una labor que promovió el doctor Jorge Vázquez, así también como el doctor Mario Molina. ¿Y cuál es la intención de todo esto? La intención de todo esto es que ustedes, estudiantes, tengan profesores de este tipo. Que lo que ustedes ven en las aulas, los conceptos básicos, van a ver dónde aterrizan, con investigaciones de alto impacto. Y eso lo hace, pues, muy bueno, ¿no? Yo quisiera ser estudiante como ustedes aquí, para que yo viera y tuviera contacto con gente como la doctora Yonath. Yo no quisiera decir más, Solamente quisiera pedir una vez más un aplauso para la doctora. Thank you, Ada. I think that you have traveling a lot of time, more than 20 hours, to be here. And this is very important for us because Ada, one of her goals is to encourage students for continue in a science career. It's very important for her to do it. And this is, I think, that one of the reasons that why you are here, Ada. Thank you very much.
Thank you. And thanks for inviting me. And I'm very happy to tell you what we are doing and even very, very recent results. So you all know the genetic code, it's sitting in DNA. You all know the DNA has only 4 letters, 4 bases. And each combination of 3 of them is coding for an amino acid that goes into a protein. I think I don't have to tell this, just to remind you. So here is DNA. The language, it means the code which amino acid goes into the protein is being here, but it's not accessible because the structure hides it. So first thing, it's being transcribed to a similar molecule called RNA, in this particular case, messenger RNA, that has the same sequence. RNA is also 4 letters. 3 of them are exactly like in DNA and 1 little bit different. It's important, but not chemically. And then this can be translated by ribosomes to growing proteins. This was known when I was a young student, younger than you. Not much, but I was younger than you. But it was not known how. I wanted to understand how. And this is what I'll tell you today, how and what we can do with this knowledge. So ribosomes that, just to remind you, ribosomes that exist in every living cell in many numbers. They have two main functions. One is the decoding, and the other is the peptide bond, formation of the peptide bond. The peptide bond is the bond that connects between amino acids. All amino acids, two of them are bound through a peptide bond. These two jobs are performed almost the same in every living cell, in bacteria, in cockroaches, in flowers, in fish, in people, in animals like lions. There is a huge number of ribosomes that function in each living cell. Even in bacteria, they can reach 100,000 when bacteria grows in the log period. And for instance, in human, as an example, in the liver, millions of them, 4 to 6 millions in each cell. And they are working all the time, making proteins all the time. They function very well. They work continuously and they function very well, very fast and with hardly any mistake. You see, the mistake rate is, there is a controversy, but it's really not important, 1 of 500,000 or 1 of 1,000,000. They can make 40 peptide bonds in a second, and I want you to look at me. I was the fastest in the class when I was second year. This is because I was very poor and I had many jobs, so I had to finish the lab fast. And we had to make 1 peptide bond, a single one, not 40, a single one. Took me 6 hours, and I was the fastest. So they can make, they can make 40 in a second. It means a protein that has 200, 300, 400 amino acids can be made in a few seconds, in less than a minute, and with hardly any mistake. So chemistry developed, and now in the lab we can make a peptide bond in 2 hours, but not 40 in a second. So after we understood how ribosomes work, it means, let me say now, in order to understand I thought so, and I think until now, in order to understand any process in nature, we need to know the structure. Structure is function. So once now that we know the structure of ribosomes, in general, we can describe them as factories that are made of two floors. In the top floor, the instructions come in. It means the messenger RNA, and they are being decoded here inside. In the lower floor, the amino acids that have to be connected to the newly grown protein are coming in by, again, of course, if it's a factory, it can be tracks, but each track is another RNA molecule, transfer RNA, tRNA. I'll talk about it in a minute. But each tRNA has one end that can decode, and the other carries the cognate amino acid. I will show it in more detail in a minute. So this amino acid, once the code for it is being decoded here, will be connected to the newly grown protein inside. I don't know to draw inside, but it's inside the factory. The protein will be larger by one amino acid. The messenger will progress by one codon, 3 nucleotides, and the track will go out empty. That's the whole story, so simple. It took us only 20 years minus 2 weeks. And it's clear that the ribosome has to have at least 2 sites for the tracks so they can connect to each other. Actually, ribosomes have 3 sites, and I'll show them in a minute. But also ribosomes have 2 subunits. The top one is the small subunit, and the lower one here is the large one. I didn't say, but I say now, this whole process is very economical energetically. It needs only little energy, and in practice, it's 2 molecules of GTP per 1 bond. So there are sites, and this, I can explain here, describe it here. So ribosome is made of 2 subunits, small and large, all ribosomes. Ribosomes did, I didn't say it earlier, did evolve with evolution. They became larger. They are 50% larger in human than in bacteria. But the pieces that do the work, the centers, the active centers are fully conserved. So we'll come back to it in a minute. And all ribosomes are made of 2 subunits, which are independent. In the cell, they are dissociated. They come together only when they have to start working. So here is the small subunit, and it accommodates the messenger RNA. And I already here hinted where are the codes. So one codon sits in a site. The codon, the first one to be translated in a site, it is called P site, peptidyl transferase center. I'll call it many times afterwards, or PTC in a short way. So the P site is here. Once this is occupied, the second tRNA can come to the A site and occupy the A site. First P and then A. This is amino-acylated tRNA. Then the bond can be made, and in each site, the tRNA sits according to the protein that it translates. So tRNA, all tRNAs in all living cells, doesn't matter how large the animal is or the organism is, they are always L-shaped double helical RNA with a loop here that is anti-codon. It means 3 codons that can interact with the codon for each amino acid, and the cognate amino acid on the other side. The other side, this side, is always CCA. You see, the whole tRNA is double helical, except for here, CCA, always, citizen, adenine, and the cognate amino acid, fully conserved. And this is the anti-codon loop. So if I can ask for dark, can we get some lower light? So if I can get lower light, you will hopefully enjoy a movie, a short movie that we made once we had once we had the structure. I checked it a minute ago and it worked. This is because of Windows 10. Windows 7 were very good. And Windows 10, but it was okay. Felipe, wasn't it okay? Oh, no, it works. So we got together with 2 students of the Art Academy in Jerusalem and we made what we think is how the ribosome functions. What you see here is bacterial ribosome. The movies, the 2 of them, I will show you one afterwards again. They are both available in YouTube, and there were thousands and thousands of hits. So I didn't put them in YouTube, somebody did, but so far, for 13 years, nobody complained. So it's probably correct, or at least nobody can show that it's not correct. So here the messenger is coming to the small subunit that takes it. Look, takes it. You see? And this is monitored by initiation factor. There are in bacteria 3 initiation factors, in higher organisms more. And this is initiation factor number 3 that monitors the acceptance of the messenger into its place. Once this is achieved, the first tRNA can come into the P site, and it is being carried by another initiation factor, number 2. Here it is, coming to its place. So the 2 factors monitor the beginning of the association with the small subunit. When they are happy, they go out, and the large subunit can reach the small one. They are interacting based on their surface and also make bridges by conformational changes. So there are 13 of them in E. coli. So now we have an active ribosome, and we look at it from the solvent side. On top is the large subunit, the lower one is the small one, and tRNAs are being brought in by other factors, elongation factors. So they come in, they are being decoded here in the small subunit, and the peptide bond is being made on the large subunit. So we took away the large subunit. You can see how peptide bond is being made, and that the newly born protein goes into a tunnel. This tunnel, I will show you it again later, is protecting the newly born protein, and it spans the large subunit. So now we put back the large subunit. You can see the whole ribosome again. Pay attention to this motion in and out of the ribosome. The red is the ribosome. 40 times a second, these very huge arms. The protein becomes larger and it starts to come out and fold directly along or with chaperones. And when there is a stop codon, instead of tRNA, there are factors like release factor or recycling factor that come in and go into the place of the tRNA, separate the 2 subunits. The newly born protein can go to where it needs, and the rest of the players are waiting for the next job. That's all. See, really, really very clever. So what you saw in the movie was small subunit and large subunit that are connected by many contacts through, you don't see now, okay, many contacts through their surface, and also by the tRNA that is here in the decoding center and here in the peptidyl transferase center where peptide bond is being made. That is called also PTC in the large subunit. And if we look at it from the side, this is small subunit, large subunit. Here peptide bond is being made, PTC, and on top is where decoding happens. If we look at the exact structure of the 2 subunits, until now I showed you some things that were not very accurately shown. They were accurate, correct, but not shown. Now you can see that each of the ribosome subunit are made of ribosomal RNA and many ribosomal proteins. The ribosomal RNA is in gray. The small subunit sediment has 30S, its molecular weight is 0.85 megadalton. It has one RNA chain with 1,600 nucleotides and 20 or 21 different proteins, you can see here. And if you see it rotating, you can identify the position where messenger binds. And its task is decoding. Large subunit with the same color code, sediment is 50S, molecular weight is 1.5 megadaltons, 2 RNA chains with 3,000 nucleotides and up to 34 different proteins. It is making the peptide bonds, but more than this, it makes the next one and the next one and the next one. It means it's a polymerase. So these are the details of the structures of both of them. And if we look at the structure of the surfaces that will see each other in the whole ribosome of the large subunit and of the small subunit, on which I marked where the AP and E site of the tRNA are bound. Actually, I didn't tell you anything about the E site. This is where tRNA exits, so it's much less important and much less accurate. So if this is the tRNA molecule, here the decoding happens, and here is the amino acid that it carries that comes to the large subunit to here. It means these two are coming together like that. And only RNA is involved in the two functions, in decoding and in peptide bond formation, only gray. So it means that the ribosome is a ribozyme. It's an RNA machine. What does it mean? It means that the cell may not trust proteins to make themselves. Yeah, it's funny, children tell me this. They say at the age of 6 or 7, but I think it's very sophisticated regulatory machinery. When you think about it a bit, it's funny, but it is. The proteins, as we know proteins, enzymes could make protein peptide bond. For them, it's nothing. But the cell, until now, biology keeps RNA in all cells, even in our cells, to make the proteins. So this means, in my opinion, regulatory machinery. It is also in good agreement with the idea, with the composition of the ribosomes that are mainly RNA, 2/3 RNA, except for mitochondria, that some of the RNA has been replaced by proteins. But the active sites are exactly the same as in any other ribosome. And it is also in agreement with the suggestion that there was prebiotic RNA-dominated world, and that RNA, which is, there are enzymes of RNA until today, 15 or 16 of them, and all of them are known to be inefficient, but nature could make RNA machine that is very efficient, like the ribosome, which I just showed you, fantastically efficient. Just to remind you what is peptide bond formation, if there is somebody here that's forgotten. So there are two amino acids, COO and NH2, and they come together and make a bond. This is the peptide bond. On this, all our life is based. In the ribosome, it happens like that. So from now on, I will keep showing A site tRNA, the one that goes to the A site with amino-acylated tRNA in blue, and the P site in green, and the ribosome in gray. So you can see here the ribosome around the positions of A and P site tRNA. Peptide bond will be made here, and there is no protein in the neighborhood, just RNA. So actually, the ribosome positions the tRNAs, the 3' ends of the tRNAs, it means the CCA amino acid, in a stereochemical situation that allows the formation of a peptide bond. That's what the ribosome does. This is where the peptide bond will be, and this is what the ribosome does. Just positioning them in place. No catalytic action there. So if you want to see it in real life, a little bit more than I showed before, this is the PTC, bacterial PTC, typical. These are all RNAs. Ignore the colors, not important. One protein is here on the shoulders, but very far from peptide bond formation. And if you want to see it together with a substrate, here is a substrate. These are results from a crystallography. It means real structures. And peptide bond will be made here. So what I said until now is that the ribosome, provides a frame from peptide bond formation, not catalytic. And our results, crystallographic results, support it. Actually, it was shown that it's not just a frame, it's a little bit, the frame is a little sophisticated. Strobel's group showed that the tRNA itself can be the catalyst of peptide bond formation, like here. So these are the two amino acids that have to be connected, and the OH of the P-side tRNA can be the catalyst for making this bond. This is all what is needed, that the ribosome puts it in this place, no more than this. If we look at how the substrates are sitting in the PTC, we see pseudo two-fold symmetry. What do I mean? Let's look at this. These are the two substrates without the ribosome, just how they sit. Here the peptide bond formation happens. And what I put here are all known substrates crystallographically from our lab and from another lab. We had no contact whatsoever. And look how similar the positions, or how exact the position is. Doesn't matter if it's much longer, the substrate, or it already grew into protein. They always sit like this. So this side relates to this, like there is a two-fold axis here, and they can rotate from one side to the other. So pseudo two-fold symmetry. When we look at the RNA diagram of the large subunit, we see no symmetry here. Or maybe one of you sees symmetry? No symmetry. When we look at the sequence, there is no symmetry. But when we look at the main chain fold, not the whole structure, just the fold of the RNA in the active site, which is here, we see symmetry. This part has two halves symmetrically related to each other. Only the structure, no sequence and no base pairing. So this part of this region is where the P side binds, and this is where the A side binds, like this. So we call it the P region and the A region of the active site, and it's altogether 180 nucleotides. So when we rotate one from here to here around this imaginary two-fold symmetry, we get such a beautiful overlap. You see what fantastic symmetry, especially for a biological material that has all L shape and so. So except for this part here that you hardly see, and this tail here, they are almost the same. If we put it, even with if the side chains, if we ignore the sequence, just purines and pyrimidines, look how nice, except for this one, which I'll come back to it in a minute. Wonderful. A region, P region. This is just the coordinates. And that's the way this machinery looked, this part looked in 3 dimensions in the ribosome today, the contemporary ribosome. So you see here the A in blue and the P in green, together with the substrates, peptide bond will be made here. Wonderful. This is the piece that I'm talking about. And this is fully conserved. at least fully conserved in the 930 different ribosomes that we looked. Maybe there are some others, but almost 1,000. So I looked at it from top, and I put one on top of the other. I put here eubacteria and archaeobacteria and eukaryotes and mitochondria, and here are the protein data bank references for it. I put one on top of the other. I could not separate them afterwards. They're exactly the same. This part, this is in the ribosome here. Fully conserved or almost fully conserved. What do I mean by almost fully conserved? Let's look at the sequences themselves. So these are the sequences of 900 PTCs. This is 100% conserved. And all of them are either 100 or 70 or 80, and very few are around 50. But still, even if they are not the same, purines are purines and pyrimidines are pyrimidines. But we call it almost fully conserved. I hope you agree with me. So the high conservation of the symmetrical region indicates that its existence is beyond environmental conditions. So we propose it to be the protoribosome. What does it mean? It means that a prebiotic bonding entity, termed by us protoribosome, is still functioning in the contemporary ribosome. So all of us are going inside us with a prebiotic machinery. And I wrote it again in an ancient way. So the RNA apparatus with catalytic capabilities functions within the contemporary ribosome, contemporary body. And if we look at it where it is in the large subunit, this is it. It is chemically connected to the two arms. You remember in the movie the two arms that moved? And it's also chemically, tRNA connected to the small subunit. So it's the center of events, the center of everything. To remind you, it looked like this. This is the ribosome, A and P, and this is where peptide bond is formed. So now that we saw it and we could more or less understand it, the first question we ask, where and when is the peptide bond formed? So when I was a student, so even a young scientist, and until today in many books, it says that a peptide bond is made in the A site. And then the newly born protein moves to the P site, and then to the tunnel. And then comes another A site itself goes to the P site, comes another A site, and the whole thing moves into the A site, and then to the tunnel. I didn't like it. I didn't see why a whole chain has to go to one amino acid. So I made, first of all, to explain myself, I made a little cartoon. You want to see my cartoon? Like here it is. So this is the A site, and this is the P site, and this is the protein. I drew it like this because I didn't have enough space for length. Now comes a new tRNA with a new amino acid. According to what was known then, this will come here, the whole thing. And then the amino acid will be put here, and then it goes back. And then it goes back. Amino acid is put here, and then it goes back. Now a new amino acid comes. It will come again here. It will be put and go back. Now a new amino acid comes. So according to this should have come to the amino acid. And then this will go up, and then the whole thing will go to the side. I didn't like it. I didn't think this whole heavy thing has to move. Don't you agree? In my opinion, this should not happen here. It should be the opposite. It should go to it. The A site should go to the protein, that newly born protein. So we try to understand this. Just to remind you, it will go here, and then it can be put. So we looked at the space available for peptide bond formation. And what did we see from top? This is the ribosome in gray. And this is an empty space between the A site and the P site. Empty space except for these two nucleotides. All empty here. Why should it be an empty space there? This is the first thing that we try to investigate. It means, to remind you, from going from here to here or making the bond, it's empty. So we thought that this empty space is used for A site tRNA to move into P site. And this requires rotation. But we all know from other studies that tRNA moves sideways. So we suggest the tRNA motion is made of two components. One, which is shift of the double helical part sideways, and rotation of the CCA around this bond that is very, very flexible, like this. Sorry, doesn't go. Like this. So what we think is that this part of tRNA goes this way, and the lower part rotates from A to P according to this arrow here. And this rotation, if we look at it in detail, is being dictated and directed by the ribosome, the walls of this empty space. And if we want to look at it in a little bit more detail, this is the volume from A to P, and this is the ribosome around it. There is no other way, actually. Only this way. And what happens at the end, as I told you earlier, we found out that the ribosome just places the two components in stereochemistry suitable for peptide bond. Look, this is what happens at the A. Stereochemistry almost fully ready for peptide bond formation. All what is needed here is 15 degrees adjustment, which can be done, I'll show you in a minute. But to check it again, we calculated what will happen if we have an A site tRNA here. We used real coordinates, we rotated by the 180 degrees, and we got it here with a stereochemistry suitable for peptide bond. So this, the ribosomologists, by the time we still didn't have structure, were aware of the fact that these two nucleotides and that these two sides of the peptide bond formation of the ribosome are important. They cannot be changed, they cannot be mutated, they cannot be eliminated. So all their attention was given to these two. But if there is a rotatory motion, what I just showed you, and the ribosome directs it, there are more than these two. This is one of these two. This is the second of these two. But actually all those on the way from the first to the second are important. So what you see here is A going into P according to our drawing, our calculations. We calculated every 15 degrees. So A starts here, and the blinking nucleotides are those that direct it. And they are all important, should be all important. Here it reaches the P site, and a new one can come, a new tRNA can come, and so on. So these are important, and these two from the other side. I'll come back to these two. And nobody supported it until there was a group in Tokyo, the group of Suzuki, that wrote me a letter and said, we found that there are 3 more nucleotides that are important for making peptide bonds. Can you explain it in terms of structure? And these were these 3. Exactly what we expected. So this was the first time that not only the 2 that are known were so important, but also this. In addition, more or less at the same time, Massa and Carle found out that the transition state between the A tRNA and the P tRNA happens in the not in the middle, sorry, at the end of the rotation, almost at the P site. So now we were sure that our suggestion is correct, and we just try to find out what is needed for it. So in our opinion, what is needed at A site is exactly correct, it's placed, and the P site has to be in the flipped orientation already, and this is indeed what we find when we look at structures. Just to show you, the transition state was found here. What you saw earlier in the empty space in real structures, it's here. Or if you look at another one in here, doesn't matter, always between the A and the P, close to the P. And the whole motion goes like this, and we called it the PTC environment. There is a front side of PTC and a back side, it just depends how you draw it, and this is the description of the rotatory motion. So additional outcome of the rotatory motion in the ribosome is that the protein, the newly born protein, comes out of the PTC in extended state, which is in good agreement with the small size of the entrance to the tunnel through which the protein goes out. Let's look at the rotatory motion again. We go from here to here, from A to P. We talked about these nucleotides. Now let's talk about these two that bulge in, that we see it here from top and here from bottom. They bulge in. And we think that we can associate at least one of them with a motor that propel the rotatory motion. So this is the one that is called 2062. 2585, the other one is busy with rejecting the amino acid. So let's not talk about it, just talk about this one. So we think that this is the motor that propel or may propel the rotatory motion. If we look at it from top at the PTC, this is this nucleotide. The others are shown here with their symmetrical mates. It means this is more or less symmetrical to this is symmetrical to that, this is symmetrical to that, and so on. And this bulges into the center and reaches the position of peptide bond formation. This nucleotide is very, 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 very flexible. This is this one. You can see here the A and the P, and this is reaching in between. Just a very simple molecule called sparsomycin could rotate it by 180 degrees, like this, from here to here. And this is sparsomycin, very simple. Also, chloramphenicol can rotate it to the other side. So we have many structures with many substrates between A and P, and this is the nucleotide, and look at them. These are all structures of it. It means the structures it takes with the different structures and different antibiotics. Chloramphenicol here, and what I showed earlier, sparsomycin. So we think that it may propel the rotatory motion. To remind you, we talk on a region within the large subunit, which is sitting here, about 4% of the size, which looks like this with the two tRNAs in a filling space presentation, or what you saw earlier, the peptide bond formation site, and the structure of the pocket that made it. In the ribosome, it's here between the large and the small subunit. It's made of two regions, A and P, and from top it looks like this, and from the side like this. This is our protoribosome. So based on the suggested existence of an RNA-dominated world, and on the finding that RNA can duplicate it and elongate itself and has catalytic capabilities, we propose that the protoribosome is the entity around which life was evolved. So if it's so important, we try to make it in the lab. So we try to construct it in the lab. If we think that it's important and it could exist as it is, maybe we can repeat it in the lab. So this is what I want to show you now. First, I have some references to convince you it's not our work, it means many other support from many other ways. One, that there was an RNA-dominated world. One, that RNA can be synthesized, or ribonucleotides can be synthesized under what is called prebiotic conditions. One, that could be possible substrate, could be amino acids bound to pieces of RNA, and RNA knows to prepare them. So as I said, we proposed that the protoribosome is the entity around which life was evolved, and we looked at possible pathways that could create it. So these are based on the existence of RNA world and that it can duplicate, and just to show you that RNA also has enzymatic activity, work not done by us. So we decided to build it, and we can build it like it's made of two parts. You remember this is the two-dimensional map of the whole large subunit, and this was the part that is symmetrically related. So we can take this part and this part. We can make many other compositions, and I just don't want to bore you with this, but at the end we should end up with something like this that could have been earlier also something like this, and this part went down. I really don't want to go into all the synthesis, although you are a chemist, so you should have understand it. I want to show you what we found. So one of our hypotheses suggested the catalytic protoribosome is a product of dimerization, because there are two parts there that are similar. This requires the existence of self-replicating and self-bonding and self-dimerizing RNA molecules. What I mean? Pieces of RNA that can be self-curving, self-interacting, and making a pocket. And the building blocks should be the building blocks that are very, very common in the RNA world and also in the ancient RNA world: stem, elbow, stem. So I want to skip these two and come back to what we really did. So first, we try to make this type of stem, elbow, stem from sequences that we thought that should fold like this. Second, we try to take the sequences in the RNA today, in the ribosome today, and cut from it the part that belongs to the P site from here, and the part that belongs to the A site from here. So now we have this part, and we could connect them. You see, we cut them out. Cut them, it means we didn't really cut. We synthesized them according to the sequence, but we could close these pieces in several ways, like here, like there, with what we call mutation or not. We could change whatever we could do. We found that no matter what we do, not all sequences dimerized. Only specific sequences from those that we designed ourselves or from the ribosome, only half of them. And in detail, the P site dimerized. You can see here dimers. You can see here, but not A site and not PNA. It means what we saw is non-uniform tendency to dimerize. We saw preference to dimerize, and we think that what we're looking at is pre-Darwinian Darwinism. It means Darwin on molecules, not on organisms. So what we think, what we can say, it seems that the original protoribosome was a dimer of two similar RNA chains. And it is conceivable that this sequence resembles the P site of the PTC. Later on, they changed to two different sequences according to their different contributions to peptide bond formation. So let's show you the results of the lab constructions. I could show that we got dimers of what we think proto-ribosomes. Let's now look at a proto-ribosome that was made according to the P site today, and possible substrates, activated amino acids, as I said earlier, they can be made. And to remind you what is the reaction to amino acids get together and you get D-peptide. In ribosomes, the ribosomologists like to use one amino acid as puromycin and the other real, because here one can put dyes or other ways to see it. We worked more than 9 years on this. And recently, in February this year, we got the first bond. So now we have a working proto-ribosome. We are very excited about it. We didn't really finish showing everything about it because the postdoc that did it, Tanaya, went to have her baby, her first baby. So she stopped. It's not a pity. She will come back in a month and she will continue. But so far, what I can tell you is that the RNA chain is coming from the consensus P site sequence, 67 base pairs. It means it's only the lower part of the pocket. It was identified by Maldi. We have controls, positive and negative controls that show that this is what works. And so far, she left it for a long time because she wanted to make sure, so we don't really know how long it takes, maybe only a few minutes. So this is what we have now that I'm so excited about. What shall we do now? We have proved the existence of what could have been the proto-ribosome. What now? So now we can look at the evolution, a little bit more at evolution. I want to tell you some of my thoughts. I do it fast. So first of all, there is a logical problem. Why should RNA construct a machine to polymerize amino acids? This is creation of a competitor that is better than you. The enzymes, the protein enzymes are better than the RNA enzymes. It means to create something better than you. So we don't think that RNA decided to do it. We think that there was an RNA machine or machineries, and the amino acid that smuggled into the prebiotic world snatched it because they are so clever. So ongoing binding experiments show that RNA, the pocket can bind small RNA molecules, and I really won't go into detail, but we can show it. Where did it happen? So in many labs, they say that the world was created in ponds or in oceans or in the desert. We don't care. Any place is good. As long as it let the RNA being there and make the dimers. Actually, it can be also within vesicles, within protocells, because this will be better for the reaction. The concentration may be larger. And we found some papers that show that RNA can keep its fold and its structure within vesicles. These are the ones that I just showed, so you can see it larger. And our structure is even simpler than those. So we can also think on the emergence of the genetic code. And here we come what was made first. So the initial D-peptide could have been the substrate for formation of next peptide bonds, and so on. In this way, oligopeptides could be formed. These oligopeptides that could perform tasks that were needed in the prebiotic world, or stabilizing the machine that made them could exist, could survive. So what do I mean by important tasks? For instance, this is a little oligopeptide with 3 histidines that can carry metal. But when I talk to most of the origin of life, they said histidine in the beginning, so complicated. Well, I think that they are wrong, because histidine is a product, can be a product of a very simple skeleton of amino acids and leftovers of nucleotides that were destroyed, like here. And a professor of this university, Lascano, showed in his group, showed that histidine exists in almost every group of enzymes known today. That's the only amino acid that exists there, and in very large amounts compared to all the others. So it could be histidine. And it could be that those oligonucleotides survived. Also those that can stabilize the machine that makes them, like this, could also survive. And we can see that even in the ribosome, here is the region, it is being supplemented by proteins. Here is one protein, L2, and another one, L36, which I'll show you how nicely it is keeping together, stabilizing the machine. So this suggests that the genetic code was created by or according to its products, which were found fit and useful, therefore survived, and led to the creation of primitive original genetic code, which co-evolved together with this project. I repeat it, the genetic code co-evolved together with its products, the proteins, and together with the ribosome that makes them. I would like, I see that some people are leaving, so I would like to skip all this and go just to the bonus that I have for you. And the bonus is, let's jump it all, I did it too. The bonus is our research on antibiotics. So the natural antibiotics that are the weapons that microorganisms from one type use for interfering with cell lives of different species are targeting protein biosynthesis mainly by paralyzing ribosomes. Almost half of the antibiotics paralyze ribosomes. So when we got the structures, actually when I started, I didn't even think that I will understand that according to the ribosomes, the antibiotics that target them. But we do, all the 30 of them. So when we got the structures, can we get dark again, please? Can we get dark again, like before? Oh. We made a little movie that shows how antibiotics do it, and this can be, if it works, this can be, anyway, this can be found in YouTube also. I'm sorry. So all of you are using Windows 10? When it works? I'll get out of my mind. Well, I think I gave up. So the natural antibiotics that are the weapons, they are stopping the ribosome by binding to its active sites, like the peptide bond formation and so on. I'll show you in a minute, but just to show you how small the antibiotics are compared to the ribosomes. Bacterial ribosomes are of 2.5 million Daltons, and the antibiotics are less than 1,000. And they bind to the active positions in the large and the small subunit to the active sites, like the decoding, the hinging, the bonding, the tunneling. Don't, I'm trying again. I think that I found a way to try again. And I want to show it to you in this little clip, if it works. Yeah, it works. You see? even Windows 10. So here you see a small antibiotic called edein that goes into the decoding path and blocks it. Here messenger comes and it blocks it. That's all. No messenger, no protein. Second one is also small. It's called tetracycline, and it occupies the position of the A site tRNA. Here. So A site can come but not bind. And one tRNA is not enough for a bond. So these were two that work on the small subunit. Now I'll show you two that work on the large subunit. The first is erythromycin. That was the first ribosome antibiotic that came into use after penicillin. And it sits in the tunnel. Here, look. A little protein can be made, 5 to 7 bonds, but not more. The tunnel is blocked. And the last one that I want to show you is clindamycin, that is really clever chemically. It goes into the bond. So you remember the bond is being made here. Chop. In the bond. Isn't it clever? So, as I just said, that all ribosomal antibiotics bind to ribosomal functional sites, but they are highly conserved. And mandatory for clinical use is the distinction between the bacteria and the patient. So how does this happen in highly conserved? Sometimes, even in highly conserved regions, there are subtle differences. And I want to show you one. We look into the large subunit, into the tunnel. This is erythromycin. You see that it blocks most of the tunnel. And if we look, if we look to the pocket that is made to accommodate the erythromycin and its brothers and sisters that accompanies with made, it means if we cut the tunnel perpendicular to its long length, we see that the pocket is made only of RNA, and all this, each of them is an antibiotic. All what we show here are crystal structures. All of them bind into the pocket and block it, either very, very well, excellent, very good, just good, but block it. And all interact with one nucleotide in the walls of this pocket. Nucleotide number 2058 according to E. coli. It's adenine in all eubacteria, in all pathogens. And this is the way it binds. Erythromycin is here, and this is this adenine. I only show this one. The affinity here is very, very, very high. Now, ladies and gentlemen, pay attention to this point, and you will see the difference between bacteria and us. This is the whole difference. This is bacteria and this is us. But when there is a G here, it's too close and there is no binding. That's all. Now, you all also know that resistance to antibiotics is one of the most severe problems in modern medicine. Resistance to antibiotics is a natural process. And here you can see a delegation that went to the jungles not far from here in Venezuela. They found a tribe that never had any medicine, Western medicine and so on, yet in their microbiome, within the bacteria within their bodies, they already found genes for resistance, which means that resistance is natural behavior, and these findings suggest or show that it will be difficult to fight resistance, because it's not really dependent only on us using it. So it shows that bacteria evolved the capability to fight toxins even before we came into the game. So as I said, bacteria develop specific molecular pathways that cause resistance. I want to show you one. It's not the most frequent one, but in ribosomes it's very important, and it exists. So you remember A and G for selectivity in erythromycin, and for a bacteria, for a pathogen is disease-caused bacteria, it's not so difficult to acquire the resistance by mutation. They just mutate themselves to G. For them is nothing. And one generation of them is 4 to 6 hours, so they can try many until they find the right one. So the appearance of a multi-drug resistance, multi-drug strains, together with the minimal, actually negligible number of new antibiotic drugs that are presently undergoing development, is becoming a real global health threat. And it seems that soon we will revert back to the pre-antibiotic era, during which disease caused by parasites or by simple or severe infections like tuberculosis, which is severe, but wounds that are simple. And as the World Health Organization said, we will reach soon post-antibiotic era, and the World Bank said that in 2050, almost 4% of the global economy will be lost because of resistance. So before I go on, I want to tell you that I don't think that we can get rid of resistance fully. It's unlikely, because bacteria want to live and because bacteria are cleverer than us, at least in terms of survival. Yet, most pharma companies don't make it. And I just want to show you why pharma companies, the numbers of pharma companies' new antibiotics. So look here. Between 2000 and today, even not 4 new antibiotics, but the resistance increases crazily. So we did some modest contributions toward controlling or combating antibiotic resistance. We decided to be more clever than the cleverest bacteria. So I want to show you these ones. Until recently, most of the available crystal structures were of pathogen models. They were found to be good to explain how antibiotics bind, but not the species specificity. So we got crystals of a very resistant bacteria, Staph aureus, and compare it to normal ribosomes. And we found out that all bacterial ribosomes are similarly made. They are all, you remember, like the normal ones. But we got many more lessons. Lesson number 1, that it's possible to increase antibiotic activity. There is a company in Vienna called Nabriva, and they increased the potency of a non-antibiotic by a factor of 25. Lesson number 2, that it's only in principle so far, it is possible to increase selectivity. We found empty space in near the antibiotic binding site to this particular pathogen, which is not empty in normal bacteria, so we can use this space to increase the antibiotic. Lesson number 3 I skipped. Lesson number 4 is really important. It's beyond my expectations and it leads to novel antibiotic binding sites. So I want to skip this and to show you this. So what you see here is, is the skeleton of normal bacteria, of the large ribosomal subunit in the normal bacteria, and additions, little additions that we found in the pathogen. So actually, when we talked about possibilities of sequence differences with statistic people, they told us, don't worry, statistically it's nothing, it's less than 10%, nothing will come out of it. But when we looked at the structure, I think that this is a real feature, and this is a real feature. Actually, let's focus on one of them. So here, this part is exactly the same in the pathogen and in the model. Also here is exactly the same. But here there is an additional feature, only in the pathogen. And this can be a potential binding site for a new antibiotic. So as I said, we found some. We found actually several of them, 25 of them, and they were around here. And blocking 16 out of the 25 inhibit protein biosynthesis. It doesn't inhibit the ribosome function, but it inhibits the synthesis of the protein because of other interactions with cellular components. So these sites are not involved in primary ribosomal activity, hence no pathogen contains genes for their modification. So we made it, we are cleverer than the bacteria. So this means that so far, since there are no positions, we can design since there are no modifications for this site, we can devise new next generation antibiotics that are degradable and selective. And I didn't talk about these two properties, so I want to do it now. Selectivity and similarity. Do you know about the microbiome? The microbiome are the thousands, 10,000, thousands of thousands of bacteria that are residing in the flora of semi-exposed mammalian organs like the guts and ears and lungs and skin. These are extremely useful for our health life, but there are new bacteria. So the normal antibiotics can attack them, and we want to avoid this. Actually, selectivity also means not only about pathogens against microbiome, but also against eukaryotes. And since all our sites, new sites, are on the surface, and the surface of prokaryotes and eukaryotes are very different. Most of the differences between prokaryotes and eukaryotes are on the surface. Here you have some views of additions of the surface. So this is not a problem for us. The problem is only to avoid microbiome destruction. And lesson number 6 is the environmental and ecological consideration. And for this, I want to tell you that most of the ribosomal antibiotics are extensions of small organic molecules that cannot be digested by us. So they come out of us with the other compounds that we don't digest, and they can go into the sewage. Oh, sorry? I see. And they can go into the sewage. And afterwards, I can't understand what happened to it. Yeah, afterwards, from the sewage, they can get into the irrigation system because not not all the purification possibilities work on them. So we get them back. But our suggestion for the new antibiotics doesn't have to have this cores that are not digestible and not degradable. We can make them from materials that can be digested or better biodegraded, like oligopeptides, nucleic acids, and so on. And we can optimize them. Now we have the chemistry in our hands. We don't need natural compounds. We can optimize them in terms of chemical properties, antibiotic actions, and degradability. So in summary, the new insights obtained from these high-resolution structures of the ribosome from genuine pathogens provide unique tools for suggesting novel sites. I showed it, suggestion how to improve clinical performance. I didn't show, but I said, and better distinction between pathogens and useful bacterial species. So we think about pathogen-specific antibiotics. It means what is called the personalized medicine, but the persons are the bacteria. So then we need a drug for each and every pathogen. And we are aware of the fact that there is an economical issue. The companies don't agree to make antibiotics now, even without us asking an antibiotic for each bacteria, for each pathogen, but we hope that they will change their mind. We also know that doctors have to identify quickly which pathogen is the problem. And for this, there are lots of very, very successful efforts now in several ways, and I won't talk about it. We also know that we will need many structures. Many structures how? By X-ray crystallography, 20 years for the first structure, and 2 to 4 years for the next ones? No, we cannot do this. So luckily, recently, three-dimensional, high-resolution single particles cryo-electron microscopy came into the game, and with this we can get very good structures without short time. And I just want to show you what we did about Leishmania, that is a parasite that is carried by sand flies, and it is very prominently attacking in mainly the south side of the world, including your country and our country, and several others, India and so on. And like last year, there were 12 million reports of it. So we extracted the ribosome from there, and we determined the structure within 3 months. You can see here all the proteins and all the RNA. And I want to show you just the quality. Here are the proteins of it. Let's look at the quality of the structure, which is very, very high, and the structure of the RNA, which is very high. We can even see the magnesiums. We can even see modifications like methylation, dihydrouridine. Actually, we saw 99 modifications in the ribosome of this parasite that were not mentioned in the literature. So here is the end. Our structure-based species-specific design aimed at minimizing. We don't think that we can get rid of widespread resistance, but we can minimize it. We cannot get rid of microbiome destruction, but we can minimize it and minimize environmental hazards. So, I'm sorry, something happened in this great computer. It's really not my fault. So all what is left for me to show you now is No. Is you don't see all my efforts, huh? Is actually the credit. So first I want to ask you if you know what was first, the chicken or the egg, or in our life, the genetic or the proteins. You know? I think that I know. Neither of them. The protoribosome was first. Second, I want to thank the Weizmann Institute that let me work for many years. I want to thank the Max Planck Institute in Berlin that was collaborating with us and also set up for us a measuring group in Hamburg. The director was Dr. Wittmann that wanted really to see the structure of ribosomes, but he died 10 years earlier and was replaced by Franceschi and then by Fucini. So I had two groups that worked together, and I want to thank all of them. Although the group in Hamburg was finished in 2004, I just want to show you that it had angels, because the technical assistant said that if you're not engine, you cannot work on ribosomes. The group in Israel is still existing. You can see here some that already left, but I want to highlight those that are important for this lecture. Ilana found the symmetry, the pseudosymmetry. Miri and Chen made the first constructs of the protoribosome. And Tanaya, who is now a new mother, together with Gil, found the way to make the first peptide bond. I also want to show you Tamar. She came 16 years ago for 10 weeks. She's still around. At the day that I took the picture, she had birthday and she made the cake. Can you see the cake? I'm not sure. I'll show you. This is her cake. And it shows that in my group, ribosomes are considered sweet. I also want to thank my family that was always helping me and accommodating me and supporting. This is my daughter, who is a doctor, and her daughter, who is my granddaughter, that when she was a school kid. And when I got a prize in Paris, she wrote a speech about me, and she said, my name is Noah, Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. And she gave me a prize. And the prize is, this is the Nobel medal. For me, her prize is as important. The grandma of the year is Ada Yonath. So it's possible to be a scientist, a female scientist with fantastic abilities, scientific abilities. You could see in the slides before how many fantastic girls are in the group. Here, Anat, for instance, is running the group, Dr. Anat Bashan. She has 3 children and she is a fantastic scientist. Ella has only two children, but she's also fantastic. But I really want to show you, to tell you that even to be a grandmother is possible, as you can see. And when I asked her, Grandma of the year, which year? So she said, every year you have to reprove yourself, otherwise I take it off the wall. So the only prize that I can lose. And this is still on the wall, you can see here. Also, ribosomes became popular. Here is a carnival. Here is me. and his wife is a ribosome. And this is what happened to me. And the artist that made it, he took the structures from the internet. He's not a scientist. And look, he made it symmetrical. Small subunit, large subunit this way. And if you can see these little things here, this is where he thinks new antibiotics should be put. So thank you.
¿Algunas preguntas?
Thank you for your very interesting talk. I want to ask you, how is the peptide rotatory motion inside the exit tunnel?
So it rotates?
Yes, but
And
inside
makes
the
a
exit.
protein. No, it's not inside, it's in the PTC. The newly born protein goes to the tunnel.
Yeah, but then the exit tunnel is changing its
No.
geometry?
No, the tunnel is a tunnel.
It doesn't change at all?
No.
Okay,
Also, the PTC doesn't change.
okay. Thank you.
Okay. First of all, thank you very much for your thoughts. I'm kind of nervous, but thank you very much. I really like biochemistry, and all these things really make me I don't know, very interested in science and such things. My question is, would you say that the protoribosome was formed before even the cell
Sure, yes.
developed? Yes?
Before the cell by far.
Oh, great. And what's the purpose of having a structure that I don't know, develops proteins and such things if there's not even a general life form like a cell, like.
Yeah, it's a good question. You can ask the molecules themselves. Maybe they will answer you. But anyway, life started from something. Couldn't start from a cell already.
Yeah.
So this is a question for everything in origin of life. And what we showed is, or we think that we showed, is that we can talk about a chemical reaction that could lead to life. I'm not sure that these molecules knew that at the end they will end up with a nice guy like you are.
Thank you very much.
Oh, hi. Thank you very much for the great talk. I wonder about the similarities of the mitochondrial ribosome and the bacterial ribosome. Is there any problem with the antibiotic design? I mean, some antibiotics directed to bacteria target the mitochondria. So
All antibiotics are directed to bacteria.
So
This is the definition.
Is there a problem with the new ones you are trying to design?
I hope that there is no problem, but for this we have to finish the design. And the company has to make them, and they don't like to make because they know two things. Well, first they think that there will be resistance. Maybe we will have less resistance. And second, antibiotics are taken when one has an infection for a few weeks. And they're usually not very costly. In contrast, the cocktails that elderly people get, and they have to take it every day, they can be very expensive. And also anti-cancer and anti-Alzheimer and so on. So the companies want to make money. This is why they are companies. It's okay.
Yeah.
They really want to do it. So therefore, they don't make antibiotics. Maybe if ours will be better, I'll be able to convince them. So far, I could not. Even when I told them that people will keep dying young as Mozart, 36, for instance, and they will not have clients for their expensive drugs, even then they didn't like my suggestions or me, myself.
Okay, thank you very much.
Oh, well, thank you. Here, this way. Okay. Oh, hi. Well, I'm actually not a student of this faculty. I studied physics, so I came because I was invited. But it was really interesting for me to know all these new things. Well, I think I knew a little bit because I took biology and chemistry back in high school. But I really would like to ask you if it's true that we are also resistant, well, we also develop resistance to antibiotics because we don't finish the treatment that doctors prescribe to us when we get sick.
We don't develop resistance. It's the bacteria that develop. But our behavior can help the development. And this is when the doctors give us a prescription, and after a few days we feel better. They say you have to take it 7 days, but after 3 days we feel better because the number of bad bacteria is now reduced. We think, oh, we feel better, we don't need anymore. And then we stop. But when we stop, the bacteria that are there are already partially resistant because they were exposed to the drug. So then they jump twice, and then we have a sort of helped making resistance.
Well, first of all, shalom, תודה רבה מורה. And I have a different kind of question. I was reading your life, and I wonder how much hard was studying chemistry in the first years of the Medinat Israel.
I didn't understand the question, but I want to tell you what she said was gracias in Hebrew. Yeah, and now go on.
Well, since I always said it's hard to study chemistry, it's a hard science, it's a lot of time, but when we mix that with the first years of Israel as a country, was hard for you as a student in those years?
Yeah, why are you asking this? Only those years, my life is hard all the time.
Well, I have another question too. Why the interest in the ribosome?
Because
Why exactly?
I was not interested in the ribosome as ribosome. I was interested in the translation of the genetic code, and the ribosome does it.
Okay. Thank you very much, תודה רבה.
Le vamos a dar a la doctora Yonath un pequeño reconocimiento de parte de ustedes, ¿de acuerdo? Thanks a lot for this beautiful talk and a little recognition document.
Thank you.
For you. Thanks a lot.
I apologize for the Windows 10 problems. I still don't know how to deal with everything.
Thanks a lot. Great talk.
Thank you.
Thank you.